Hello and welcome to Chapter 21 of the AP Biology Exam Review video series. We are going to talk about the evolution of populations. So you need to know how mutation and sexual reproduction can each produce genetic variation. So here's your big reminder. Why are we talking about evolution of populations when we're starting with genetic variation? Well, it's because evolution depends on genetic variation between members of a population. If there is not variation between individuals, there cannot be natural selection. If there's not natural selection, there's not evolution. And so it all boils down to genetic variation existing between different members of the population. That genetic variation, the definition for that down here at the bottom, is just saying differences among individuals in the composition of their genes or other DNA sequences. Okay, that is what is subject to natural selection and evolution. So this is actually a diagram I didn't show you in lecture. Genetic variation at the molecular level. Well, and I've said to you guys many times in class that most mutations are bad. Well, why? Well, it's because organisms are already well matched to their environment. That's what makes them successful in the first place, is the fact that they're well matched to their environment. So mutations are almost always deleterious because they reduce the fit between the organism and the environment. Every once in a while, things go right and mutations are good, but by and large, why are mutations usually bad? Because we're already fit to that environment pretty well. So this is just the diagram that goes, it's not a particularly insightful diagram, aside from to remind you that mutations that happen in introns aren't real important because those aren't expressed, or at least as far as we know, they're not real important. And then, we can have substitutions, and because of wobble in that genetic code, we can have substitutions that either don't result in a different amino acid, and of course, that kind of variation would not be phenotypic, or wouldn't be expressed. But then we can also have substitutions that do result in a different amino acid. And so these mutations, whether they're deletions or substitutions or frame shifts or whatever, they're almost always bad because of this business about being well matched to your environment. But every once in a while, like I said, they do work out pretty well. We talked back in Unit 2, in our cell unit, about variation from sexual reproduction. And so in sexually reproducing species, sexual reproduction allows for differences or I should say variation, coming from three places: crossing over, independent assortment, and random fertilization. Crossing over, that's what's happening in prophase 1 of meiosis, and that's where we have exchange of homologous portions of non-sister chromatids of homologous chromosomes. And so that was a whole lot of vocab words, but essentially all that's saying is we get our homologous pair, one from mom and one from dad, and these two are sister chromatids, and these two are sister chromatids, and this is your homologous pair. While when we're talking about crossing over, what we're talking about is the same portions that are swapping places, but they're on chromatids, but they're not sister chromatids, because the sister chromatids are this, and this is one sister chromatid, and then this, and this is the other one. And so when we swap those two things, that's what we're saying. Non-sister chromatids on homologous chromosomes swap places. That's crossing over, prophase 1. Independent assortment, that's in metaphase, in metaphase 1, where remember that in metaphase 1, the ants go marching 2 by 2, and so to add to this, okay, so here we are aligning, so that's how we know that it's metaphase. Well, let's say this one is mom, and this one is mom, and this one is mom, and this one's mom, and then these are dad. Well, you eat one of each pair of your homologous chromosomes come from mom and one comes from your dad. And so then as you undergo meiosis in order to produce your own gametes, how your parents' chromosomes align on that metaphase plate is independent of every other pair, which is how come we end up with dad. This one, how this one lined up is different from how this one lined up. Okay, so that's independent assortment. Random fertilization is just referencing the sheer number of gametes that are produced, and it's in literally the millions. And which of those gametes are successful in actually being fertilized is random. You don't get to pick which egg and which sperm actually goes on to produce your children. That is a random process. And so these three features of the cell cycle and of reproduction are what are introducing variation into sexually reproducing species, in addition to the mutations that we had earlier. And of course, mutations are consistent in producing variation for all species, whether or not you are producing sexually or asexually. You're going to remember that some phenotypic variation is not heritable. So just because things look different doesn't mean that that is a heritable change. Here we have two worms that we have the cute, the fuzzy one, and then the one that looks like a stick. Two caterpillars whose variation is phenotypic, but has to do with their diet. And so chemicals in diet I forgot an S. Chemicals in diet can actually cause phenotypic variation, but that's not heritable because it's the chemicals that are causing the change in appearance, not the genetics. And so the genetics that's what is a heritable impact. Hardy-Weinberg, you have to know the 5 conditions, and here they are. The 5 conditions. We're going to remember that when in equilibrium, a population is not evolving. And we're going to talk about what that means in another slide here. But the 5 conditions are that there are no mutations, that all mating is random, so we don't have any mate choice or sexual selection going on. There is no natural selection, so any differences in survival don't exist. A massively large population. This is so that number 2 works out, because remember, the larger the population, the less susceptible genetic drift is. And so we have random mating and an extremely large population size to kind of allow for the statistics to work. And then no gene flow, so nothing coming and going from the environment. Alleles aren't coming into the population, alleles aren't leaving the environment. So if it's kind of like, okay, we have to have this population that is perfectly unchanging. And when all 5 of those conditions are true, we are going to be in Hardy-Weinberg equilibrium. So you need to know how to use that Hardy-Weinberg equation to calculate allele frequencies to then test whether the population is evolving. And if the population is evolving, then you go back to those 5 criteria that we just looked at in order to figure out which one of those 5 is being violated. And so you have to look at your allele frequencies to say, okay, yes, the population is evolving. If so, which one of these 5 conditions are we violating? Or no, the population is not evolving, which means all 5 of these conditions must be true. So here's your Hardy-Weinberg equation. Okay, you're going to remember that your P and Q references allele frequencies, but then this one, P squared plus 2PQ plus Q squared, each term here represents the frequency of that particular genotype in the population. So this is homozygous dominant, homozygous recessive, and then heterozygote. Of course, in standard Mendelian genetics, these 2 terms would all express the dominant condition. So you want to keep that in mind as well. Now, what I want you to understand is that no matter what, P plus Q is always going to equal 1. It's a frequency. That's like saying I have a deck of 52 cards. Every single one of them is red or black. And so if I add up the proportion of my red ones to the proportion of my black ones, I'm going to have 100% of my cards. It's not like all of a sudden there's a blue card in there somewhere and that is allowing us to not have P plus Q equal 1. So I don't care whether the population is in equilibrium or it isn't. P plus Q must always equal 1. So what do you actually look at to figure out if the population is evolving? What you look at is, okay, what is the P and what is the Q, and is that changing over periods of time? So let's suggest for a moment that you do some calculations and you find out P is equal to 0.4 and Q is equal to 0.6. 10 years down the line, you look at it again and P is 0.5 and Q is 0.5. This indicates that there is evolution occurring. Then you go back to those 5 conditions and you see which one's being violated. If, in contrast, 10 years after collecting your original data, you go back and you find the same numbers again, then you can say, okay, this population is in Hardy-Weinberg equilibrium for this particular gene, and indeed it is not evolving when it comes to this particular trait. You must know what effects genetic drift, migration, or selection may have on a population and analyze data to justify your predictions. This comes back to that computer lab where I had you guys manipulate all the different variables and look at the lines on the graph. That's what we're looking at here. So genetic drift, we're going to remember that genetic drift is random, and it is most significant in small populations. Okay, and so in the example I used in class was we have a field of flowers, a moose tromps through the woods and kills a whole bunch of flowers, and where he stepped just happened to be that he stomped all over, in this case, the red ones. And then, you know, later on, the moose tromps through again, and this time he kills everybody except the red ones. And so it's when we're dealing with these really small populations where a moose tromping through the woods could result in a massive swing in allele frequencies, that's genetic drift. Do we really have moose tromping through the woods that are stepping only on red flowers or only on pink flowers? Well, no. But that's just to give you an illustration to say that random impacts can cause allele frequencies to change. And in a large population, that doesn't tend to have an overall impact on your P's and Q's. But when you're only dealing with a handful of organisms, or in these cases, 10 organisms, every time one of them dies, you're losing 10% of those alleles, and that can result in major shifts in the P's and Q's. How do we see this in, examples aside from the moose tromping through the woods? The founder effect is a good example of this. If you have a whole bunch of birds living on an island, there they are, and then we have this distant island way over here that's just outside of the reach of how far those birds can fly. But then all of a sudden, not one, but two of them get stuck in a snowstorm or a windstorm or something, and two of them make their way all the way over. Well, those two can start a new population over here. We call that the founder effect. Those two are then going to be the only two birds that are able to contribute genetics to the population on this island. And there they are. Well, the thing is, if we had 100 birds over here and we only got 2 birds over here, the chances of those two being able to completely represent the genetics of those 100 is nearly impossible. And so that's one of the reasons that a small population is so impacted by genetic drift. That's the founder effect. We also have what's called the bottleneck effect. And the bottleneck effect is just saying, well, if a population undergoes a huge reduction in size very, very quickly, and that reduction in size really has nothing to do with the genetics or the relative fitness of that population, then that is going to also greatly impact your P's and Q's. And so bison is a good example of that. In North America, before Columbus sailed the ocean blue, bison were living really coast to coast. And then we came over, Europeans came over and exploited the bison for all sorts of things, but mostly profit. And as a result, they were nearly wiped out. And so now that major shrinking of the bison population has resulted in our modern day populations of bison that are descendants from just a small handful of survivors. And so those survivors, they had to go on, and now we have plenty of bison again, but the gene pool of those bison is very, very, very small. Modes of selection, these diagrams tend to be pretty straightforward. And so here what we see is we have directional selection, disruptive selection, and stabilizing selection. And for each of these, we're starting with the population. This is just any kind of phenotype is along the bottom. Here they're showing you mouse color. That's fine. Directional selection is just selecting for one way or the other, where the organisms at one end of this bell curve are more susceptible to, let's say, predation or something like that. And so then if the white ones all get eaten off, we will evolve in this direction. We can have disruptive selection, where the extremes are favored, but this medium one is not. And then we can have stabilizing selection, where the two extremes are selected against, but then that central phenotype is favored. And you could go back to your notes and see the examples I gave. I think I gave baby birth weight, human birth weight as an example of stabilizing selection. If you're too small or you're too big, you're likely to have health problems, is the one I gave you there. So this is just 3 different modes of selection. We can also have heterozygote advantage. This is where we're actually selecting for the heterozygote. The example I gave you guys had to do with sickle cell malaria. sickle cell anemia that had to do with malarial resistance. And what was happening, or what still is happening, is that this central portion of Africa, sub-Saharan Africa, is very susceptible to malarial infection. And the trick is that if you are homozygous dominant, let's use M's. If you are homozygous dominant, then you are totally normal. You do not have sickle cell anemia, but you are susceptible to malaria. If you have little m, you have sickle cell anemia and you're probably not going to survive to reproductive age. However, if you are heterozygote, what that means is that you do not suffer the effects of sickle cell anemia, and you also don't suffer the effects as badly for malaria. And so this is the ideal place to be if you're living in sub-Saharan Africa, genotypically. The trick is, what's happening is that is preserving the existence of this deleterious allele in the population. And so in other parts of the world, this particular allele has died off long ago. It's not nearly as prevalent. However, in the sub-Saharan Africa, you can see the frequencies of these alleles are much higher than most of the rest of the world, which falls into this category of less than 2.5%. And so that's the heterozygote advantage. Sometimes being a heterozygote is the best condition to be in. We also have what's called frequency-dependent selection. And that's where what genotype is selected for, what phenotype is selected for, is completely dependent on what is already in the population. And that's where frequency-dependent selection comes in. And so just the example I gave you guys in class was, these guys are scale-eating fish. This is the fish that they eat the scales of, and these guys aren't stupid. They learn that if they're always getting bit on the right side, that they need to protect their right side, and then if they're always getting bit on the left side, to protect their left side. So the way that this particular predator keeps them on the move is that there is a selection for the mouthedness, either being right-mouthed or left-mouthed, in the population. And so if you track over time what's happening with being the right-mouthed or left-mouthed, you see a shift in which one is more dominant in the population. And that way, this guy never gets to learn, oh, I should always protect my right, or I should always protect my left. Lastly, the last one for this video, you need to know why natural selection cannot fashion perfect organisms. Okay, so four reasons why we don't have perfect organisms on the planet. First, selection can only act on existing variations. So it's not like painting a picture where if you don't like it, you're going to crumple it up and start over. Selection can only exist on what is currently in the environment. And so new advantageous alleles can't just arise on demand. All that can happen, we have to remember this, all that can happen is that individuals can live to reproduce or die trying. And it's that event that dictates what the population is going to look like. And so if existing variation for a particular trait doesn't exist, the new allele isn't going to just be produced. And to that end, evolution is limited by what we call historical constraints. Evolution operates on the traits an organism already has. So this is a outlandish example, but let's suggest that humans were going to develop wings. We were going to evolve wings. Well, if I were going to have wings, I would not want to lose my arms, because I want to be able to fly, but I still need to be able to, write and dial the phone and carry a coffee cup around and all the other things I do. Well, the trick is that it would be a compromise. Chances are, if I were going to fly, it would be my forelimbs that would become my wings, and I would no longer have my arms. And so that would be the compromise, is that I could get something, but then I would lose something. So in that, I would be imperfect. An organism has to do lots and lots and lots of different things. And so because we have such flexibility with our usage of limbs, well, that makes us prone to some kind of damage as well, sprains, torn ligaments, and whatnot. And so all of these things are reasons why modern day we don't have perfect organisms. Now, to then take it one step further, the planet is constantly changing. And what is advantageous right now may not be advantageous in the future. And we can even think about that with regard to seasonality and change. We have a winter that's really, really wet, And then we might have, for the next 4 years, winters that are really, really dry. And just that basic environmental shift over a couple of years is enough to have different advantages for different phenotypes. And so when we talk about, chance, that should say chance, not change. Chance, natural selection, and the environment are all interacting. The bottom line is that what's advantageous right now, this month, this day, may not be advantageous 5 years from now. And even on very, very short periods of time, these things that are selected for are going to be changing over time. So that's the end of this video. Thanks for watching. I will see you in class.